Hello and welcome to the 15th episode of the Innova Biosciences webinar series. Today's webinar, Antibody Purification, What You Need to Know to Use Antibodies Effectively, will discuss the various methods available for purifying antibodies from different sources and explain why it is vitally important to understand how your antibodies have been purified, to know what you can do with them, either within assays or for further processing such as conjugation to dyes and enzymes. Our speaker today is Dr. Andy Lane. During his presentation, Andy will discuss sources of antibodies, different methods of purifying antibodies, why purified antibodies are not all the same, how to purify antibodies easily without specialist knowledge or equipment, and considerations for conjugating antibodies to enzymes, dyes, and nanoparticles. There will be a question and answer session at the end of the presentation. Please submit your questions by typing in the questions box in your control panel, which is circled in red here. Feel free to send questions throughout the webinar and we will answer as many as we can in the Q&A session. Any remaining questions will be answered by email in the next few days. And that's the basics laid out. Now please welcome Andy.
That's great. Thank you very much, Claudina, and good afternoon to everybody for our webinar about antibody purification. As Claudina said, I'd like to start just very briefly talking about different sources of antibodies to put some of those into context. And this slide just summarizes probably the most common sources of antibodies that people will come across. The first is animal serum, really from any species that's commonly used to raise polyclonal antibodies, perhaps typically rabbits or sheep or goats, etc. And then with monoclonal antibodies, we have two sources. Ascites, which was a source probably really from the early days of monoclonal antibodies and is certainly less commonly used now, especially in European countries. And cell culture supernatant, where the hybridomas are grown in vitro in bulk format. And both of these are used as common sources of monoclonal antibodies. Other common sources of antibodies, and becoming increasingly common, are eggs for the avian antibodies, particularly chicken, IgY, which is becoming increasingly popular, and also a number of bacterial expression systems as recombinant antibodies become more common in use around the world. The key thing about each of these is that they obviously all have the specific antibody that you're interested in, but that antibody is contained amongst other proteins, other antibodies itself, lipids, etc., and lots of other components. And it's all of those things that you need to purify antibody away from, and that's what we'll be talking about today as we go through this. I'm going to be talking about a number of different methods of purifying antibodies. I probably won't cover every single method that is available, and I may not go into absolute detail on all of these, but I hope to give you a good summary of the various most common techniques. The ones I'll be talking about are summarized on this slide. So I'm going to start off talking about fractionating serum by globulin precipitation, move through ion exchange and size exclusion chromatography, talk about protein A, G, and L affinity chromatography, and then also antigen affinity chromatography. I'll take some time to go through each of these over the next few slides. I'll start with globulin precipitation, which is really the most basic form of antibody purification, and remarkably was actually first published in a format in 1899 by James Atkinson in the Journal of Experimental Medicine. He could actually show that he could separate the antitoxic components of a horse antiserum using magnesium sulfate. This was remarkable at the time because people's understanding of antibody responses, etc., was actually not very great. Of course, things have moved on since then, and rather than using magnesium sulfate, now the most commonly used process is to use ammonium sulfate, taking advantage of the principle of salting out. Basically, when you add ammonium sulfate at increasing concentration, various proteins begin to precipitate out of solution. When you get to about 50%, the globulins or the immunoglobulins are precipitated out of solution, and many other proteins, including the protein that is of highest level as a contaminant, being albumin, that remains in solution. So effectively, by adding ammonium sulfate at a particular concentration, you can purify, or at least partially purify, immunoglobulin away from the major contaminants in the serum that you're working with. It's relevant to say that this is a very simple and gentle procedure, but it does only provide a partially pure preparation, especially if you're starting from a typically complex mixture such as serum. This step is therefore often combined with further purification using one of the other methods that we'll discuss in just a moment. Indeed, it's often the first step for purification with some of these, so you're getting a partially purified preparation moving on into these other methods. The next technique I'd like to talk about briefly is ion exchange chromatography. Ion exchange chromatography uses a positively or negatively charged resin to bind proteins based on their net charges in a given buffer system. So typically you take a complex mixture and add to a column containing the resin that you're interested in, and you do that in a certain set of buffer conditions. In that particular set of buffer conditions, different proteins will bind to the resin and some proteins will not bind to the resin. For antibody purification, you set up buffer conditions so that immunoglobulins will bind to the resin and most other proteins will pass through. So for instance, albumin will not bind in a certain set of conditions, will move through the column whilst immunoglobulins are left attached to the column. You then change the buffer conditions either on a stepwise or a gradient basis, and the different proteins are released from the column and can be separated and purified. Ion exchange chromatography is probably more often used to purify polyclonal antibodies rather than monoclonal antibodies, who tend to be purified more by protein A and G, as we'll discover later. However, with ion exchange chromatography, there are some advantages that might be applicable for repeated purification of the same monoclonal antibody. Remember that every individual monoclonal antibody is unique in terms of its sequence and therefore in terms of its overall charge, and it will therefore bind and be released from an ion exchange column under a very specific set of conditions. Therefore, if you want to repeatedly purify the same antibody, you can define the set of conditions for the preferential purification very consistently, and therefore you can have a very optimized situation for that purification. There are all sorts of ion exchange resins available, but probably the most widely used is something called DEAE cephorose, which many of you may be familiar with. This is a diagrammatic representation of ion exchange chromatography, and you'll see as we move from left to right, on the left-hand side we have a column in the starting buffer, and we have two substances to be separated. We have the green and the yellow proteins just about to be added to the column. As they pass through the column, they bind to the resin, and then you're about to add gradient ions in the different shades of blue to the column. You add the pale blue first and pass those through, and these change the ionic conditions such that the yellow protein is released while the green protein remains attached to the column. As you increase the concentration of the gradient, now looking at the dark blue circles, that actually releases the green protein. So you can separate the yellow from the green simply by increasing the concentration of these ions within the buffer. You can then clean the column up going back to the starting buffer, and that column can then be used again if that's what you need to do, and you can purify various different antibodies taking this approach. Moving on from towards size exclusion chromatography now, This is a method where the name very well describes exactly how things work. You have separation of complex mixtures on the basis of size or molecular shape. You actually undertake a molecular sieving process. So the solute, which can be an antibody in this particular instance, passes through a packed bed of resin. The separation of that antibody from other components depends on the different abilities of the various molecules present to enter the pores of the bead-based stationary phase. In effect, this means that large molecules which cannot enter the pores are excluded from the column, and that means they pass through the column very quickly. Smaller molecules can enter the pores to varying degrees, and therefore they're slowed down. They take a longer course through the column, and they move through it more slowly. The very small molecules, such as the buffer and salt components, fully permeate the column, and they actually elute at the very last step. You tend not to use size exclusion chromatography for primary purification of antibodies, other than perhaps for IgMs, which you may be familiar with as being pentameric forms of IgG. In effect, they're very large molecules, around about 900 kilodaltons. And size exclusion chromatography, often in combination with ammonium sulfate precipitation, may be used for IgM purification. However, size exclusion chromatography is also a valuable so-called polishing step following purification by other methods. So if you haven't quite achieved a perfect purification using one of the other methods, a run down a size exclusion column may actually give you a final step of purification that can be very helpful. There are a number of chromatography resins that are available, and you have to choose these based on the size of the molecules that you want to separate. So ultragel columns come in different sizes of ACA 22, 34 and 44, for instance. And in the Cefidex range, S200, S300 and S400 resins are very useful for purifying antibodies at various stages. I did just want to mention a particular use of size exclusion chromatography, and that's in the area of so-called desalting columns. Desalting columns are generally small volume columns that are used to undertake a buffer exchange or to remove small size contaminants from a buffer. So you might want to change an antibody from one buffer to another. By passing this down a particular size of size exclusion column, you can change the buffer because those small molecules are held up on the column whilst the larger molecules, the antibody, moves straight through. And that allows you to do a buffer exchange. The most commonly used is the Cefidex G25 column, often in prepared form, which is a PD10 column. So this is just a specialist form of size exclusion chromatography, which many of you may actually use in the lab without thinking of this as being a particularly complex form of chromatography at all. There are a couple of figures here explaining size exclusion chromatography on a diagrammatic basis. And these really show the same thing, but in different ways. So on the left-hand side, you see a column which is packed with a resin, which are these blue circles. Based on the shape of the resin, you can see there are three sizes of proteins present. A small protein, which is the red molecules, an intermediate green, and a larger purple protein. You'll see that the red molecules actually get caught up in the gaps within the resin, and therefore they move through more slowly. They take a longer route through the column. Green is not caught up so well, it won't actually move into the pores very well, and purple actually just moves between the resin, doesn't get in between the resin particles at all, and moves through most quickly. If you look at this on the right-hand side, you'll see the three added to a column at the same time. Purple moves through most quickly, then green, and then red. And if you're collecting those off of the end of the column, you can get a very good separation of these proteins based on their different sizes. I'd like to move on now to the protein A or G and L chromatography. Now, I'm sure the great majority of you will be familiar, certainly with protein A and protein G chromatography. These are probably one of the most commonly used forms of antibody purification that is now being used. In their native form, each of these proteins are expressed by different bacteria as part of the bacterial defense mechanisms against the mammalian immune response. In effect, certainly with protein A and protein G, they simply get antibodies to bind, shall we say, in reverse to the surface of the bacteria, which means that the effector mechanisms of the host immune response are somewhat blunted and the bacteria are protected from destruction. All three of these proteins bind to mammalian immunoglobulins via constant regions on the antibody. Proteins A and protein G bind to the Fc region of antibodies, and protein L actually binds to kappa light chains. And we'll talk a little bit more about protein L and kappa light chains as we go through the next couple of slides. Although initially native versions of these proteins were used for purification, nowadays almost exclusively recombinant versions are used, and these are immobilized to various matrices, allowing people to purify IgG by what is in effect a form of affinity chromatography. Diagrammatically, you'll see the antibody molecule in front of you now. You'll see that protein G and A bind to the tail or the Fc part of the antibody, and protein L binds to the light chain. And as I said, remember this binds only to kappa light chains, not to lambda light chains. For mouse monoclonal antibodies, that really isn't much of a problem. Around about 90% of mouse monoclonals actually express kappa light chains, so you have a very good chance of protein L binding to them. But remember, if you're purifying human antibodies, the ratio is more of a 60 to 40 of kappa to lambda, and therefore protein L will only be purifying a proportion of human immunoglobulins. When using any of these, you simply add your sample to the particular column that you have in a suitable binding buffer. You need to consider both the pH of that buffer and the ionic strength. Generally, you bind at a neutral or possibly even slightly alkaline pH. You wash the column, and because this is an affinity column that really should be binding only antibody, all other proteins and materials should literally wash straight through. That then leaves you with a column which in effect should only have IgG bound to it. Your next step is to carry out a low pH elution. You can use various different buffer formulations for this, and they actually may differ depending on the isotype of the antibody being purified. I've got a couple of tables in a moment and we'll focus on that a little bit more. It may well be important to know the basic formulation of your antibody and the formulation of the elution buffer for downstream processing. You may use different buffers. They may be glycine-based, for instance, or citrate-based, and that may be important for you later on. As you've been eluting the antibody with low pH, most people will then add a high pH buffer to neutralize the pH and therefore to protect the antibody from any damage that might occur at low pH. This is often carried out with tris buffer, and that will take the pH back towards pH 7 very effectively. In many cases, you can use the antibody immediately from there, but you do need to be aware then that that antibody will be in a buffer that contains the elution buffer and tris, and that might also have an effect on how you can use that antibody further downstream. This table actually shows just a summary of the different proportions and different ways in which protein A, protein G and protein L are derived, and some of the key factors that they have. So if we start with protein A on the left hand side, this is actually derived from Staph aureus. It has 5 immunoglobulin binding sites and has an optimal binding pH for immunoglobulin of around about 8 to 9. This means that you can elute antibodies at a range of pHs. Antibodies that bind strongly may need to be eluted at pHs as low as pH 3, but some antibodies can be eluted with a pH actually as high as pH 7. And this is isotype dependent, and it's very useful to know what the isotype of the antibody is that you're purifying. Protein G, it actually comes from Streptococcus. Protein G doesn't have as many binding sites for immunoglobulin, and it has an optimal binding pH that is somewhat lower, around about pH 5. Somewhat associated with that means that the elution pHs for protein G are rather lower than for protein A, and that of course could be considered to be slightly harsher conditions for your immunoglobulin. So typically pH 2.5 to 3 are typical elution pHs from protein G. I'm going to skip to the protein L column, so second from the right. This protein L is a more recently identified protein that can be used for affinity chromatography and immunoglobulin purification. It comes from an organism known as Streptococcus magnus. It has 4 binding sites for immunoglobulin, a relatively neutral optimal binding pH, but again quite a low elution pH. You'll also see within this table that I've mentioned some joint recombinant protein columns. So we have columns that may have either protein A and protein G attached to them, or recombinant versions which are actually fusion proteins of protein A and G together, or even protein A, G and L together. The advantages of these are that they provide a rather broader spectrum of purification than the simple protein A or protein G itself. So people will have their own preferences. Many people will actually start using protein A as being the slightly more gentle version for purification because the elution pHs are a little higher. But with some antibodies, as we'll see in just a moment, protein G or protein L may be more appropriate. In this table, I've summarized just some of the binding strengths of different antibodies and different isotypes to protein A, G and L. If you look around on the internet, you'll find much more extensive tables of this sort, looking at other species immunoglobulins from all sorts of different animals and unusual sources. But I've stuck really here to human, mouse and rat for the monoclonal sources, and then goat, rabbit and sheep as the most commonly used polyclonal antibodies. Without reading through every single box, I think what you can see if we look at protein A is that it's generally quite good for human immunoglobulins, with the exception of IgG3. It's quite variable for mouse immunoglobulins and is not really very good at all for rat immunoglobulins. Goat and sheep antibodies also really don't bind very strongly to protein A. Moving to the column for protein G, you'll actually see that this actually has really stronger binding than protein A to virtually all of these different species and isotypes. It's a little bit weak with rat IgG1, but even that is better than the binding of protein A to rat IgG1. So you can see that protein G may have some attractions for people who need to purify a range of different antibodies. and need to know that their resin is likely to bind to them quite well. Protein L, on the right-hand side, you'll see has strong binding to human, mouse and rat immunoglobulins. But as I said before, remember it only binds to kappa light chains, and if your monoclonal or source doesn't have a kappa light chain, then you won't be able to use protein L for that purification. Also note that protein L isn't so good for some of these polyclonal sources, really not good at all for goat and sheep immunoglobulin, and not as good for rabbit antibodies as it is for the rodent monoclonals. So having talked about protein A and protein G and L as a form of affinity chromatography, I'd like to move on to perhaps what is the gold standard for antibody purification, and that is antigen affinity chromatography. In this case, you prepare a specific chromatography resin actually by binding the antigen that the antibodies are specific for to that resin. So you have a column on which the antigen of your interest is bound. You then add your source of antibodies, they may be a polyclonal serum, for example, to that column, and only the antibodies that are specific to that antigen will bind to the column. All other contaminants, and that includes any other antibodies that are present, will be washed away. I'll come back to that a little bit later because it's important to recognize the difference between antigen affinity chromatography and protein A and protein G chromatography. As with protein A and protein G chromatography, the next step in antigen affinity chromatography is to change the buffer conditions so that you elute the specific antibodies from the column. Again, this is generally done by utilizing a low pH. Typical examples of the uses of affinity chromatography are in preparing species-specific secondary antibodies. So those of you who use secondary antibodies may see various descriptions of them, but Affinity Purified represents a high-quality antibody that has been purified using this technique. Affinity chromatography is also widely used to purify polyclonal antibodies that have been raised against peptide antigens. And a form of affinity chromatography using columns that are specific for epitope tags is often used in purifying recombinant or phage display antibodies. Again, in this diagram, we see various steps in antigen affinity chromatography, and here we're actually starting at the top with the preparation of your gel matrix. So your matrix is the purple square, and your ligand, so this is the target protein, is combined with the column. And there are various kits that enable you to make these columns yourselves. You have pre-activated matrices, and you can bind ligands to them. Once you've made your resin, you can then add your sample to that resin, and the yellow sort of bracket-shaped molecule here, which really is in effect an antibody, will bind to your ligand, and any impurities, as I said, that may include other antibodies, will pass through the column and be moved away. And then the last step at the bottom, you change the pH conditions and your antibody is released and can be purified. So these are just some of the common methods for purifying antibodies. And as I've alluded to during a couple of the slides, there are some things that you need to understand about the differences between these techniques. I'm just going to talk about a few of these now with a few slides that I've entitled "Why Purified Antibodies Are Not All the Same." If you're buying antibodies from commercial companies, and there are many tens of thousands of these available, they may often be sold as purified. What does that really mean in effect? I've listed down here some of the things to consider when you're looking at more detail at these antibodies. Possibly one of the most obvious is to think about the concentration of the purified antibody. This at least will give you an indication of how much you can dilute the antibody for use, and therefore an antibody that is presented perhaps at 1 milligram per mil may be of use for more applications than something that is presented at 10 micrograms per mil. You may also want to consider preservatives and stabilizers. And I've put a comment here saying, have they added anything back into the preparation after purification? So for the last few slides I've been talking about how to purify antibodies away from other proteins. Once you've done that, you end up with a purified IgG, but if it's sold and kept at relatively low concentrations, the stability of that antibody may not be too good. You might therefore find that a stabilizer, and often this is bovine serum albumin, has been added back to the antibody. So you have a purified antibody, but it actually has high levels of albumin present in it. If you want to do anything to that antibody involving steps such as conjugation, that albumin will be a real problem to you. You also need to consider, for some applications, the buffer components. And I talked earlier on a little bit about the elution buffers that you use from protein A or protein G, etc. These may contain contaminants that are relevant to you, and I'll go into more detail about that on another slide. I'll also talk a little bit more, as I've alluded to, about the level of purity, and also just consider some unexpected contaminants that you may not actually think about too carefully. Finally, you might be interested, depending on your application for your antibody, in knowing whether there is any endotoxin present. Endotoxin is particularly important if you're working with live cell systems, and some manufacturers will measure this and declare the levels to you. So let's just think about some of these other main factors coming up. I'm going to talk about the buffer components first. So the final buffer that your antibody is in can have a significant effect on what you can use that antibody for. So you might actually use a glycine-containing buffer to elute antibody from a protein A column. Glycine will actually inhibit many conjugation reactions, and we'll talk about those a little bit later. You might also have sodium azide that's been added as a preservative. That's great unless you want to incubate that antibody with live cells, and sodium azide may actually kill those cells. You also need to be aware that different buffers might be suitable for different forms of conjugation. So buffers that are suitable for undertaking conjugation to proteins might not be suitable for conjugation to nanoparticles. I just wanted to mention the importance of dialysis in making purified antibodies. So as I said, when you've purified something on protein A, you might be happy to leave the antibody in the buffer that it was eluted in, that has been neutralized, say with tris. So you might have an antibody that is effectively in a glycine buffer with tris added, and it's at a neutral pH. If you're actually buying a commercial antibody, you'll probably find that most have been dialyzed into phosphate buffered saline, and many of you would want to do this yourselves. It's a very neutral buffer, isotonic buffer that's useful for the widest range of applications. You undertake this by dialysis, but you do need to make sure that your dialysis has been effective. Just to give you an example of how to ensure that dialysis is effective, we're going to take an example where you've got 10 mils of antibody and it's in a buffer containing 100 millimolar glycine. You want to effectively remove that glycine altogether, and by altogether here I mean glycine down to a level as low as 1 nanomolar. If you want to do that in one step, you have to dialyze your antibody against 1 million litres of PBS, and I'm absolutely certain none of you want to do that. Alternatively, simply by changing the 1 litre dialysis 4 times, you can achieve the same effect. So that's 4 litres of PBS instead of 1 million. This really underlines that changing the dialysis buffer is very important. But do remember, if you only dialyze that 10 ml of antibody against 1 litre of PBS, your glycine will still be present and it will be there at 1 millimolar, which is a relatively high concentration for things like bioconjugation reactions. Another aspect I just wanted to talk about briefly is so-called total IgG against specific IgG. Sometimes people aren't really aware that when you raise a polyclonal antibody, even after several rounds of immunization, only approximately 10% of the immunoglobulin that is present will be specific to the antigen that you're interested in. So 90% of the IgG that is in the serum is IgG against various other factors that the animal may have raised antibodies against during its lifetime, or just normal background IgG with no particular specificity. If you take a step to purify all of that IgG, therefore only 10% of the IgG that you get will be specific for the antigen of interest. The only way that you can get IgG that is nearly 100% specific is to use antigen affinity chromatography. If you use affinity chromatography using protein A or protein G, that actually only gives you total IgG. It looks 100% pure if you analyze it on SDS page, but remember that only 10% of those IgG molecules at best will be specific for your antigen. These differences will actually give you significant differences in activity. So remember that the working dilution of an antigen affinity purified antibody could well be tenfold higher than that of a protein A or G purified antibody. It's well worthwhile looking at antibody data sheets to see exactly what you're getting. You might also sometimes see that companies supply only small amounts, perhaps 50 microliters of one antibody, and maybe half a mil or 1 mil of another antibody, and the prices look quite similar. You might therefore think you're getting not very good value for the 50 microliters, but if it's antigen affinity purified, it's likely to be approaching tenfold as potent as your protein A or G purified antibody. I also just wanted to mention a particular aspect that people working with tissue culture supernatant may not consider, but also may be quite important. So here I'm thinking about bovine IgG. So by definition, if you purify a monoclonal antibody that's been raised in or produced in cell culture supernatant, you would assume that this would be a 100% pure. This is the only antibody being produced by the hybridoma cells, so there's no other antibody to get in the way. Just by putting that question, is that right? You'll probably be aware that I'm going to say not necessarily. You need to be aware that if your cell culture media contains either fetal bovine serum or perhaps worse, newborn bovine serum, there might be significant levels of bovine IgG within that serum. If you use protein A or protein G to purify your antibody, you may well get co-purification of bovine IgG with your monoclonal antibody. You can avoid this by using a mouse IgG specific purification system based on affinity chromatography, or of course by converting all of your hybridoma cultures to serum-free media. That's a step that some of you will realize is not maybe as straightforward as it might be. I'd also just like to mention that if you use ascites, obviously you avoid the presence of any bovine IgG, but you will have normal mouse IgG present, which also co-purifies on a protein A or G column. So these are just some of the things that you need to be aware of when you're thinking about purification. I'm going to move on a little bit now to thinking about some simpler ways of purifying things, and then very briefly to some aspects of conjugation. So I've put a picture up here of actually a relatively simple purification system in a lab. Many of you will have seen purification systems and columns that look much more complex than this, but also many of you will look at this and think that this is a fairly complex and expensive system. But purification doesn't have to be anywhere near as complicated as this. As part of the range of conjugation products that Innova Biosciences has available, we've developed a purification range of products alongside these. This AppSelect range takes the same approach as our approach to conjugation, really to try to simplify things as much as possible. So with these products, you avoid the need for expensive pumping systems, recording systems, etc. All you need is often a centrifuge, and you'll be able to perform the purification that you need. These kits are all designed at small volumes, particularly in relation to being able to prepare antibody for conjugation. Obviously, that purified antibody can be used for a whole range of other applications as well. We have a flow chart here which just takes you through some different steps. I'm not going to go through every single step of it, but just take you through some of the key parts. So when you start at the beginning, you ask the question, has your antibody been purified? And if not, if it's a serum product or a tissue culture supernatant, you move to the right-hand side and you'll see that Innova offer a range of different products that will enable you to purify very simply your antibodies, either from culture supernatant or serum or ascites fluid. Perhaps your antibody has already been purified and it's suffered from one of those things I've mentioned earlier, that the antibody company that's made it, for a very good reason to maintain stability, has added something like BSA to the product. So we start moving down the left-hand side, realize that we have BSA present that we'd like to remove. You can undertake this using some of the purification systems showing in the middle, or you can skip right to the bottom for a new product that we have, for a product that is called the BSA removal kit, which I'll discuss in another slide in just a moment. Note the BSA removal kit does what it says in this context, but won't remove gelatin if that has been used as a stabilizer. And then on the left-hand side, we have a product, the Antibody Concentration and Cleanup Kit, which is really designed to help you concentrate antibodies to a convenient level, or to remove small buffer components that may interfere with whatever you want to do with your antibody. So this will remove tris, sodium azide, glycine, etc. in a very simple and quick method with very good yield. I've used the AppSelect range with a couple of slides just to show some examples of purification, and this first one picks up the question that I mentioned a couple of slides ago about contamination with bovine IgG. Here we actually have the rat purification kit as part of the AppSelect range, and this will be for preparing and purifying a rat monoclonal antibody that has been prepared in cell culture. If we take the second column from the left-hand side, this is a control rat IgG, and next to it is bovine IgG. Now these two run at slightly different molecular weights, which makes this easy to show you on this SDS page gel. If you use protein G agarose, so seeing in the middle, protein G actually co-purifies bovine IgG and rat IgG, and although that looks like a single band, it's quite a blurred band in the middle there. If you move to an AppSelect rat resin, and you look second from the right, this is the eluted fraction looking for bovine IgG, and you'll see that the AppSelect rat resin does not purify any bovine IgG, but on the very right hand side you have a pure population of rat IgG. So you can avoid co-purifying bovine IgG with the AppSelect kit. And we have kits available for both mouse immunoglobulins and rat immunoglobulins that can help you avoid bovine IgG contamination. The other SDS page gel that I'd like to show you just shows the effect of using our BSA removal kit. And this is really designed to remove BSA from an antibody that has been purified and had BSA added as a stabilizer. So again, moving left to right, you'll see second from the left an immunoglobulin control around about 150 kilodaltons, BSA at just over 55 kilodaltons, and then an antibody product that contains IgG with BSA as a stabilizer. So you'll see both bands present. Using the BSA removal kit, the IgG after separation, second from the right, shows you purified IgG from which BSA has been removed. And on the very right hand side, the BSA component of that has been shown. So you can separate those two out very neatly, and you'll also see on the right hand side that there is very little immunoglobulin left in the BSA fraction. So you also have very, very good yields. The BSA removal kit will also allow you to undertake a buffer exchange and remove components such as glycine if you have them at the same time. I'd just like to spend a couple of slides talking about conjugation products, and in particular how these work alongside purified antibodies and some of the things you need to think about. Many of you may be familiar with the Lightninglink conjugation technology from Innova. This is the world's fastest and easiest to use conjugation technology. It only takes 30 seconds to label your antibodies with dyes, enzymes. We have a range of over 50 labels of this type. So 30 seconds hands-on time and a reaction that can be complete in 15 to 20 minutes with some kits and 3 hours with our standard kits. We also have conjugation technology that moves into different areas. So we have particle conjugation technology with our Innova Coat Gold range and our recently released Innova Gold high quality gold nanoparticles. And we also add to these ranges with our Funderlink range of kits that allow you to conjugate oligonucleotides to antibodies for applications such as immuno-PCR. So with these range of kits, you can see that it's very simple to conjugate antibodies to a range of enzymes, dyes, particles, oligonucleotides, etc. There are some different things, however, that you need to think about when conjugating your antibodies using these kits. All of them are very simple, but you need the protein, or the antibody in this case, in the right format. We've talked about the concentration of antibody and ways in which to get your antibody at 1 milligram per mil or a little bit higher. We've talked a little bit about purity, and if you're undertaking conjugation, you really do need a purified antibody. You need to ensure that other proteins have been removed, and as I mentioned with BSA earlier on, make sure that they haven't been put in again afterwards. You may need to go back through a removal step if so. Buffer formulation is perhaps one of the most important things to think about. Make sure that your buffers avoid amines such as glycine, as well as materials such as DTT, mercaptoethanol, etc. Once again, just consider how your antibody has been eluted from protein A or protein G, and if it's been thoroughly dialyzed after that elution. You also need to think about the buffer formulations of the antibodies, and note that different kits, especially conjugation to nanoparticles, can have different buffer requirements and purification methods than for conjugation to enzymes. Because of this, we've recently extended the AppSelect range, so we now have an AppSelect range which is defined for antibody labeling, and a new range which complements AppSelect called AppPure. And these are purification kits that are particularly for purifying antibodies for conjugation to nanoparticles. So we offer different purification systems depending on what you want to conjugate your antibody to. Once again, this is aimed at making your life as easy as possible from the conjugation perspective. I hope this has been helpful in reviewing some of the different antibody purification methods, and also for some of the different things that you have to consider that may not be top of your mind when perhaps purchasing purified antibodies. We've had a few questions, I think, coming in during the webinar, and I'm just going to pass back to Claudina now for a couple of comments, and once we take a look at some questions, and we'll begin to answer those in just a moment.
Okay, thank you very much, Andy. It is indeed time for our Q&A session now. So there's still time to submit questions. Simply type in the questions box as shown in the picture on the slide, and Andy will now answer as many as possible in the time allocated, and any remaining questions we will respond to by email in the next few days. Okay. Our first question today is, you mentioned that antibodies purified from ascites may be contaminated with normal mouse IgG. Can you explain that more, and is it a major problem?
Okay, that's an interesting question around ascites. Many of you will be aware that actually ascites production is now quite difficult in many parts of the world, but it's still relatively common in certain areas and probably as an overall method of monoclonal production. Ascites is produced when you inject hybridoma cells into the abdomen of a mouse, and the antibody that's produced is contained in a matrix that is effectively very similar to mouse serum. It contains many other proteins, and those will actually include normal mouse immunoglobulin. And when you purify the antibody, especially with a technique something like protein A or G, the normal immunoglobulins will co-purify with the monoclonal. If your level of monoclonal antibody is relatively high, then the presence of the normal mouse immunoglobulins may not be too significant. They'll be quite low in percentage terms. But if your monoclonal antibody level is low, contamination from normal immunoglobulin may be quite high. You can't actually identify normal immunoglobulin contamination on an SDS page gel, as the antibodies all run in exactly the same place. You need to use a technique called serum protein electrophoresis to identify the monoclonal antibody against a background of normal immunoglobulins. That technique separates out the antibodies based on charge rather than on size.
Okay, so here's another question. In some of my applications, I use unpurified antibodies without any problems. Can I get better results by purifying the antibodies?
Okay, thanks for that question. Indeed, there are some applications in which unpurified antibodies give very good results. For example, cell culture supernatant may be used in immunohistology staining and is very effective. Of course, in those cases, you always need a secondary antibody detection system, and you will find that purified antibodies may give you more options for direct labeling or maybe multicolor labeling. They may also provide you with more consistent results from batch to batch of material.
Okay, and our next question is, how can I purify antibody fragments such as Fab2?
Okay, so I'm going to assume that this is Fab2 that's been prepared by a sort of enzyme digestion from IgG, and the approach I would take to that would be to use size exclusion chromatography with an appropriate column. If you're using one of the ACA columns, an ACA 44 column would probably be best.
Okay, and our next question is, how can I remove endotoxin from my antibody?
Okay, for endotoxin, I would actually say that the best thing to do is not to introduce endotoxin into the preparation in the first place. That's actually much easier than many people expect. It really comes down to keeping all of your buffers and the purification columns you're using very clean, making sure you're using purified water and very clean glassware, etc. Removal is possible using affinity columns that are designed to bind endotoxin. One example is the antibiotic polymyxin B, which can be used on an affinity column. In my own experience, these are actually quite difficult to use and provide fairly poor yields of antibody at the end, and they're not always especially effective at removing the endotoxin.
Okay, and our next question is, when I've used purification columns in the past, I always seem to get antibody at really low concentration. How can I concentrate the antibody, or can I get the columns to deliver antibody at higher concentration?
Right, so when using any column, there are a few basic principles that allow you to maximize the concentration of antibody that you're recovering. Basically apply the smallest sample possible, therefore at the highest concentration. Run the column slowly and use the smallest size column that's appropriate for your sample size. So if you apply samples at high concentration in minimum volume, that also gives you maximum resolution on the column and also the highest concentration when the antibody is eluted. I think the question also asked about concentrating antibodies. Various ways of doing this, but probably the most common now are to use spin cartridges, which you can find in various different sizes. Use those with a centrifuge and make sure you've got an appropriate molecular weight cutoff to concentrate your antibody from the buffer that they're already in.
Okay, thanks, Andy. Our next question is, can I just use the BSA removal kit instead of protein A to purify my monoclonal from cell culture?
Okay, the answer to that one is unfortunately not. We've designed the BSA removal kit to remove BSA that has been added as a stabilizer to a previously purified antibody. In a more complex environment, such as a cell culture supernatant, you will succeed in removing much of the albumin present, but you'll probably co-purify other proteins with the antibody as well, so we wouldn't recommend it for that use.
Okay, and our next question is, what about hydrophobic interaction chromatography?
Right, that's certainly one of the techniques that I haven't included in the main webinar. In effect, it's a technique that you can use to polish antibodies that have been purified by other methods. It relies on the interaction of antibodies with a hydrophobic ligand on a column matrix. In some ways, you could think of it as being similar to ion exchange chromatography. In hydrophobic interaction chromatography, proteins bind to a column in high salt conditions and tend to be released in lower salt conditions. It may be used to follow up with ammonium sulfate precipitation, as I mentioned right at the beginning of the webinar, as a second purification step after that stage.
Okay, I'm afraid we only have enough time for one more question, but like I mentioned previously, we will respond to any outstanding questions by email in the next few days. So our last question for today is, I heard that protein G also binds albumin, so have avoided using it. How can I overcome this?
Okay, thanks, Claudina. So the question about protein G and albumin, native protein G from Streptococcus actually does have an albumin binding site, and you'll see reports of that in various places. However, that has been engineered away in the recombinant forms of protein G that you'll find that are available on the market for antibody purification. So I really wouldn't have any concerns about that being a problem.
Okay, thank you very much, Andy, and thank you everyone for sending in those questions. So again, please bear in mind that this webinar is recorded, so we will upload it to our YouTube channel in the next few days so that you can always come back to it. And you can find our YouTube channel at youtube.com/Innova Biosciences. And if you would like to receive a PDF copy of the presentation or a link to the video recording of the webinar, then please contact me. You can find my contact details on any of the webinar emails that you've received. So thank you very much for joining us today. We hope that you enjoyed the webinar, and we hope that you can join us again in September.